So let's walk through this complicated slide. The G protein coupled receptor here can respond to two types of activation or signaling. So in one case, upon binding of the agonist, it can associate with the G protein coupled receptor. We then have an exchange between GDP to GTP in G protein, which is a heterotrimeric protein. That in turn results into dissociation of the G protein into several subunits, some of which can either go on to activate adenyl cyclase and so form CAMP from ATP. And that in turn results in downstream signaling. The bound GTP also can get dephosphorylated to GDP again, and then the subunit gets recycled back. Here, we see here the alpha subunit. Alternatively, upon binding of G protein, we also have this dissociation resulting in binding to ion channels and activation of, in this case, of the potassium ion channel. Once again, the beta and gamma subunits then get recycled and reassembled in an intact heterotrimeric G protein bound to GDP, then it can interact with a new receptor. An important thing to remember here in blue, we have what happens upon binding of the agonist. So the gray helix, which is transmembrane helix 6, changes its conformation and becomes a blue one upon binding. And that conformational change also makes it able to bind to GRK, which in turn allows for phosphorylation of the receptor itself. Upon phosphorylation of the G protein coupled receptor, then we have a binding of arrestin, which ends up inactivating the receptor. And it does that by several mechanisms, either by desensitization or by internalization of the receptor into the endosome and then destruction through degradation. So this intrinsic dichotomy serves to regulate the concentration of the receptor on the cell membrane. So another thing to consider is that although the general topology of the G protein coupled receptor is the same, so they all have 7 transmembrane helices, what changes is the ligand binding site. So that will depend on the specific receptor type. And so specifically for monoamines, we have a pocket at the top of the transmembrane helices. And that includes adrenergic and muscarinic receptors, which we'll cover in due time when we talk about neurotransmitters. In part B, we have binding of peptide hormones, which basically involves, besides the top of the transmembrane helices, also involves the extracellular loop and the N-terminus chain. Other hormones bind to the extracellular loops and N-terminal chain exclusively, so that there is no transmembrane involvement. And finally, glutamate, which, as we just saw, it's another possible neurotransmitter, binds here on the N-terminal side. So let's talk about the various type of G protein subunits. So several type of G proteins can be activated by the same type of receptor. In general, they all share the same topology, so they're heterotrimers, and they're located intracellularly but associated with the membrane. They bind GDP. When the receptor is activated, then we have a change in shape, and so GDP is exchanged for GTP, and that in turn causes the G protein to dissociate from the channel, fragment, and be released. And so this again results in a signal amplification because several G proteins are activated by one molecule of ligand. And then basically the alpha subunits of various types can carry this message to the next stage. So let's talk briefly about the types of um, actions mediated by these alpha proteins. So alpha S regulates the adenylate cyclase, so basically makes facilitate the formation of cyclic AMP from ATP, and we know that CAMP functions as a second messenger. One example of that will be protein kinase A. So basically, CAMP regulates the activity of protein kinase A. Alpha Q regulates phospholipase C, and so regulates the metabolism. Other type of alpha subunits can regulate the cGMP. They can also open calcium and potassium channels. All right, the next class of receptors I want to cover is the so-called tyrosine kinase linked receptors. In this case, the protein really has a dual role, so it's a receptor as well as an enzyme. Upon binding of the messenger, we have a change in conformation, and that opens up the active site of an enzyme, which remains active until the messenger departs. So the enzymatic action caused by binding of the messenger multiplies and amplifies the signal. So tyrosine kinase are typically formed by one transmembrane helix, and they typically dimerize, as we'll see in a second. The extracellular component contains the N-terminus and the binding region for the ligand. And then the intracellular domain contains the C-terminus as well as the catalytic region here. What kind of reaction is catalyzed by tyrosine kinase? As the name indicates, the reaction catalyzed is the phosphorylation of tyrosine. So tyrosine contains a phenol group, so an OH, an alcohol on the aromatic ring, and in the presence of this enzyme of magnesium and of ATP, we have a transfer of a phosphate molecule from ATP to tyrosine. And that is involved again in downstream signaling. A typical example of tyrosine kinase linked receptors is the growth factor receptors, so, such as the epidermal growth factor receptor. We'll talk about this kind of receptors again when we talk about anti-cancer agents, because generally this kind of receptors can be involved in cancer. What happens then when the EGF, so the epidermal growth factor, binds to the receptor? So binding causes dimerization of the receptor, and then what we have is that tyrosine kinase active site on the intracellular portion is opened up and activated. So in the presence of ATP, what happens is that each side of the receptor phosphorylates the other side. And so, in turns, phosphorylation of the receptor causes a downstream signaling cascade that we'll see in a second. So what happens then? Well, what happens is that, in other mechanism, first, the ligand binding and then the conformational changes causes dimerization and then once again, phosphorylation. From here, ghrelin 2 binds to the phosphorylated part of the receptor, and then in turn recruits RAS, which is an oncogenic protein, and exchanges GDP with GTP in RAS, and that in turn causes activation of RAS and downstream signaling. So what happens exactly? Activation of RAS inactivated all these proteins by phosphorylation, typically, and in the end, that results in gene transcription. And so these kind of receptors are used to mediate cellular growth, both in physiological and in pathological conditions.